5 грантов имени Кудайбергена Жубанова. Зарегистрировано 85 патентов. Учеными факультетами передано в библиотеку по 85 книг. Студентами реализовано 85 стартап-проектов. В 2020 году Жубанов Университи перелистнул 85-ю страницу своей истории, чтобы с новыми целями идти вперед вместе с вами.
Dear students and colleagues, my name is Aaron Chikanover, and I am an Israeli physician and biologist working at the Faculty of Medicine of the Technion, which is Israel Institute of Technology in Haifa, Israel. I happen also to be a laureate of the 2004 Nobel Prize in Chemistry for discovering the ubiquitin-mediated proteolytic system. I intend to participate and to give a talk at the Central Asia Nobel Festival series of lectures, and I invite you all to participate, to inspire us and to be inspired. But mostly, I want you to learn from this participation one lesson. People like us who made important discoveries are human beings exactly like you are. We have 2 hands, 2 eyes, 2 legs. We are simple human beings, and if we could do it, you can do it as well. So all the best, and hope to meet you all in the upcoming festival of Nobel laureates in Kazakhstan. Take good care of yourself and stay safe.
Welcome to the Central Asia Nobel Fest in 2021. This is John Mather, Senior Project Scientist for the James Webb Space Telescope, working at NASA in Greenbelt, Maryland, the United States. I'm interested in the whole history of the universe from the Big Bang to now, how life became possible on our own little planet circling an ordinary star, how we can protect our planet, and how far we can go. I hope you will find the Nobel Fest a fascinating adventure.
Hi, I'm David Spergel. I won the Breakthrough Prize in 2018, and I'm excited about speaking at the Central Asia Nobel Fest on my work and others on cosmology. Over the past 20 years, we've learned that atoms, the stuff that makes up us, makes up only 5% of the universe. So I'll be telling the story of how we probe the dark matter, the dark energy, and look back to the beginnings of the universe and learn how structure and galaxies and everything else emerges. Look forward to seeing you soon.
Hello, I'm Christopher Haken, 2018 Breakthrough Prize winner. I'm very excited about the upcoming Central Asia Nobel Fest lecture series. I hope you'll join us.
Nobel Fest.
I am working as Center of Chemical Engineer at KB2 since 2015. It is a great pleasure to welcome to our center. Study at our center provides students new knowledge, new technology. Our Center of Chemical Engineer has trained more than 500 chemical technologists. Our degrees lead graduates to successful lives and careers as scientists, specialists, and scholars. Our bachelor program is the best in Kazakhstan by independent Kazakhstan agency. We have international accreditation. Our programs licensed by international agency. I can say that KB2 is the #1 in Kazakhstan. It will be glad to see you in KB2.
События 2020 года. Пандемия, самоизоляция, Локдауны. Да, но не только они. 2020 год – это плюс 15 тысяч жубановцев, новые лаборатории и кабинеты, новые возможности Smart University, создан автономный образовательный портал с собственной системой прокторинга, увеличено количество услуг Smart RSU, проведена неделя цифровизации. Достижения ученых. Учеными выиграны гранты на 160 миллионов тенге. Под руководством профессора Кванышбека Шункеева разработан учебник по физике для школьников Казахстана. Открыт диссертационный совет на соискание степени PhD по филологии. Защищена диссертация по математике. Успехи студентов. Волонтерские организации и школа волонтеров стали лучшими проектами области. Проведен международный студенческий семинар «Перспективы развития студенческого самоуправления в эпоху цифровизации». Участники стартап-академии выиграли гранты на свои проекты почти на 3 миллиона тенге. Международное признание. Университет стал соорганизатором международной конференции QS. Жубанов University впервые вошел в международный рейтинг QS University Rankings и ECA и занял 351 место среди лучших вузов. Год Абая. Имя и наследие Абая стало ближе тысячам казахстанцев. Проведено множество мероприятий и конкурсов. Опубликовано несколько научных трудов. Книга Абайтану передана во все школьные библиотеки области. И, конечно же, 2020 – это юбилейный год университета Жубанова. Проведено 85 благотворительных мероприятий. 503 человека награждены юбилейными медалями. 85 педагогов получили премии от областной профсоюзной организации работников образования и науки. Выпускники Актюбинской области стали обладателями 85 грантов имени Кудайбергена Жубанова. Зарегистрировано 85 патентов. Учеными факультетами передано в библиотеку по 85 книг. Студентами реализовано 85 стартап-проектов. В 2020 году Жубанов University перелистнул 85-ю страницу своей истории, чтобы с новыми целями идти вперед вместе с вами.
Dear students and colleagues, my name is Aaron Chikanover, and I am an Israeli physician and biologist working at the Faculty of Medicine of the Technion, which is Israel Institute of Technology in Haifa, Israel. I happen also to be a laureate of the 2004 Nobel Prize in Chemistry for discovering the ubiquitin-mediated proteolytic system. I intend to participate and to give a talk at the Central Asia Nobel Festival series of lectures, and I invite you all to participate, to inspire us and to be inspired. But mostly, I want you to learn from this participation one lesson. People like us who made important discoveries are human beings exactly like you are. We have 2 hands, 2 eyes, 2 legs. We are simple human beings. And if we could do it, you can do it as well. So all the best, and hope to meet you all in the upcoming Festival of Nobel Laureates in Kazakhstan. Take good care of yourself and stay safe.
Welcome to the Central Asia Nobel Fest in 2021. This is John Mather, Senior Project Scientist for the James Webb Space Telescope, working at NASA in Greenbelt, Maryland, the United States. I'm interested in the whole history of the universe from the Big Bang to now, how life became possible on our own little planet circling an ordinary star, how we can protect our planet, and how far we can go. I hope you will find the Nobel Fest a fascinating adventure.
Hi, I'm David Spergel. I won the Breakthrough Prize in 2018, and I'm excited about speaking at the Central Asia Nobel Fest on my work and others on cosmology. Over the past 20 years, we've learned that atoms, the stuff that makes up us, makes up only 5% of the universe. So I'll be telling the story of how we probe the dark matter, the dark energy, and look back to the beginnings of the universe and learn how structure and galaxies and everything else emerges. Look forward to seeing you soon.
Hello, I'm Christopher Haken, 2018 Breakthrough Prize winner. I'm very excited about the upcoming Central Asia Nobel Fest lecture series. I hope you'll join us.
Nobel Fest.
I am working as Center of Chemical Engineer at KBTO since 2015. It is a great pleasure to welcome to our center. Study at our center provides students new knowledge, new technology. Our Center of Chemical Engineer has trained more than 500 chemical technologists. Our degrees lead graduates to successful lives and careers as scientists, specialists, and scholars. Our bachelor program is the best in Kazakhstan by independent Kazakhstan agency. We have international accreditation. Our programs licensed by international agency. I can say that KBTO is the #1 in Kazakhstan. It will be glad to see you in KBTO.
События 2020 года. Пандемия, самоизоляция, локдауны. Да, но не только они. 2020 год – это плюс 15 тысяч шибановцев, новые лаборатории и кабинеты, новые возможности Smart University, создан автономный образовательный портал с собственной системой прокторинга. Увеличено количество услуг Smart Арсу, проведена неделя цифровизации. Достижения ученых. Учеными выиграны гранты на 160 миллионов тенге. Под руководством профессора Конышбека Шункеева разработан учебник по физике для школьников Казахстана. Открыт диссертационный совет на соискание степени PhD по филологии. Защищена диссертация по математике. Успехи студентов. Волонтерские организации и школа волонтеров стали лучшими проектами области. Проведен международный студенческий семинар «Перспективы развития студенческого самоуправления в эпоху цифровизации». Участники стартап-академии выиграли гранты на свои проекты почти на 3 миллиона тенге. Международное признание. Университет стал соорганизатором международной конференции QS. Жбанов University впервые вошел в международный рейтинг QS University Rankings и ECA и занял 351 место среди лучших вузов. Год Абая. Имя и наследие Абая стало ближе тысячам казахстанцев. Проведено множество мероприятий и конкурсов. Опубликовано несколько научных трудов. Книга Абайтану передана во все школьные библиотеки области. И, конечно же, 2020 – это юбилейный год университета Жубанова. Проведено 85 благотворительных мероприятий. 503 человека награждены юбилейными медалями. 85 педагогов получили премии от областной профсоюзной организации работников образования и науки. Выпускники Актюбинской области стали обладателями 85 грантов имени Кудайбергена Жубанова. Зарегистрировано 85 патентов. Учеными факультетами передано в библиотеку по 85 книг. Студентами реализовано 85 стартап-проектов. В 2020 году Жбанов University перелистнул 85-ю страницу своей истории, чтобы с новыми целями идти вперед вместе с вами.
Good afternoon. My name is Jeanette Kunz. I am a faculty at the School of Medicine at the Nazarbayev University here in Nur Sultan. And I will moderate the first talk of this afternoon session. And this is already the last day of Nobel Fest, which is a collaboration between international institutions, businesses, and also hosted by the Inclusive Development Foundation and Kazakhstani universities. And this is the second edition of Nobel Fest. And I think we had again the privilege to listen to a whole panel of distinguished scientists and businessmen that discussed the research and innovative endeavors. And I hope this will be a tradition from now on and will raise interest in science and innovation in this region. And for this afternoon session, it is my particular privilege and pleasure to introduce a fellow Swiss scientist, Dr. Michael Hall, who is our first speaker in this last session today. And Dr. Michael Hall is a professor at the Biozentrum at the University of Basel in Switzerland. And this also happens to be my alma mater. And in fact, Michael's laboratory was really where my own scientific research began. So before I start, I wanted to give a little bit of background on the speaker and the topic of today's talk, both of which are actually quite cosmopolitan. So Michael was born in Puerto Rico. He grew up in Venezuela and Peru. He then moved to the United States for his studies, and he finished his PhD at Harvard University. He then moved on to the Institut Pasteur in Paris, in Europe, returned shortly to UCSF in San Francisco in the US before he moved in 1987 to Basel, where he still works at the Biozentrum as a professor and also former chair of the Department of Biochemistry. And the scientific story that Michael actually will summarize, I think, today, also started in a very exotic place, namely Rapa Nui, also known as Easter Island. And it started with two natural drugs that were isolated from Rapa Nui, and which Michael happened to work on for many, many years to come. And by studying drug action, he actually made a whole series of seminal discoveries, both in yeast and mammalian systems, that relate to how cells control growth. And it is also related to the control of cell metabolism. And this is implicated not only in normal development and in aging, but is also very important for a number of human diseases, including cancer, cardiovascular disease, diabetes, as well as immune disorders. And by studying these drugs and making his seminal discoveries, Mike really went from the beginning to now drug discovery and treatment in the clinic. And for his work, his distinguished work, he obtained a number of prizes. I think if I looked at the whole series, there is now one outstanding, but I only want to mention the 2014 Science Breakthrough Award, the 2017 Lasker Award, and last year, the Sherberg Award, which he was awarded jointly with Dr. Sabatini from MIT, who also works on the control of cell growth. And before I take any more of the precious time, I want to hand over to our speaker. And at this point, I also wanted to mention to our students that they should ask the questions via the chat function, but I think they can also ask directly via Zoom and maybe even during the Zoom session. So please, Mike, it's your turn. Thank you for joining us today.
Okay, thank you, Jeanette, for this nice introduction. Let me share the screen. Okay, well, it's a real pleasure to be with all of you. It's actually the second time. My only regret is that I'm not there live to meet you in person. I hope that happens once. This is actually the second time I was invited to speak in Kazakhstan. The first time was several years ago by Das Sarbasov, and unfortunately something arose which I couldn't do it then. So hopefully on the third time I'll actually be able to visit all of you. Many of you probably know Dr. Sarbasov. I don't know if he's listening, but if he is, I'd like to extend some warm greetings to Das. Okay, so what we do is we work on Tor signaling, in particular the role of Tor signaling in cell growth control. And we do this in yeast and in mammalian cells. And what I'd like to do today is give you a very long and largely historical introduction to Tor signaling. I'm not going to tell you our latest results or actually even show you any data because this is not a specialized audience, a specialized Tor audience, so I thought you'd benefit more from a general introduction to the topic. Okay, so I'll tell you essentially just the story of Tor. So this story began back in 1991. This is a condensed overview of the Tor field. This over here is the very first ever model of Tor signaling published. It's probably one that Jeanette Kunz recognized. It's actually one that she published in a paper that she wrote while she was in the laboratory. I'm actually a little embarrassed. I never told Jeanette this, but I'm actually a little embarrassed we published this model because it's not very useful. It says Tor exists and very little else. There's question marks upstream and downstream. And much of what we have here, for example, G1 progression and PI, phosphatidylenosytol, turned out to be wrong as we learned more about Tor signaling over the years. I don't know if many of you remember the science cartoonist called Gary Larson, but this reminds me of a Gary Larson cartoon more than something you'd find in a science publication. So today, the picture is considerably more complex. This is even already out of date. And this increase in complexity over the last 30 years has been driven by this exponential increase in publications in this area, and this continues to go on beyond 2012. And what's driven this increase in publications is that Tor turned out to be both fundamentally important and clinically relevant, much more so than we suspected when we first stumbled across it back in the early 1990s. So let me take you back to 1991, and by the end of the talk today, I'll have you at a state of knowledge which resembles something more like what you see over here on the right of the screen. So here's the timeline of the Tor field. The story actually started before 1991, started in 1965, because this was when, as Jeanette mentioned, this is when scientists went to Rapa Nui to collect soil samples. And what they were doing is they were prospecting for exotic microbes which would produce novel secondary metabolites, which they could then develop into drugs. In particular, they were looking for antifungal drugs. They did isolate such a microbe from the soil in Rapa Nui. They did, in fact, isolate a metabolite from that microbe, bacteria, and they began to develop as an antifungal. However, as they were developing as antifungal, they made the observation that it had the undesirable side effect of suppressing the immune system. And of course, suppressing the immune system is the last thing you want to do to someone who's suffering from a fungal infection. So it was quickly rejected and almost forgotten as an antifungal. Years later, though, it was rediscovered, now exploited for the very property which led to its rejection early on, the fact that it suppresses the immune system. It was redeveloped, repurposed, but now as an immunosuppressive drug, and it was part of the important evolution in medicine of the development of transplantation surgery. So it was one of the few drugs which made possible the transplantation of organs. If you go to Rapa Nui, there's actually a plaque which commemorates this picking up of this soil sample for the isolation, which led to the isolation of rapamycin. And here's that plaque. It's in Portuguese, and it says, "In this place was obtained in January of 1965 a sample that allowed not only the isolation of rapamycin, but also inaugurated or ushered in a new era in transplantation medicine." So having worked on rapamycin for 30 years, I always wanted to go to Easter Island or Rapa Nui to pay homage at this site where rapamycin originated. And I actually did this recently. It was one of the last things I did before the COVID pandemic. So one of the last trips I ever took. And here we are. This is the site. This is that same stone you saw on the previous figure. And much to our disappointment, this is where the plaque used to be. Someone in the years since had unfortunately stolen the plaque. But we went to this site as soon as we got off the airplane, went straight to it. This was our local guide. And this is another well-known person in the Tor field, Nahum Sonnenberg, who was on this trip with us, as well as some other Chilean scientists visiting Rapa Nui, which is part of, which belongs to Chile. So it was a big disappointment not to see the plaque, but we at least got to see the place where this whole story started. This is what rapamycin looks like, this natural secondary metabolite that is secreted by soil bacterium from Rapa Nui. As I told you before, it was developed as an immunosuppressive, which facilitated organ transplantation. But now it's even perhaps more important as an anti-cancer drug, based on the fact, as you will hear, rapamycin blocks cell growth, and it's even used in the treatment of restenosis after angioplasty, so in the treatment of cardiovascular disease. So this one drug, this one molecule, which had this rather humble beginning in the dirt in this very isolated island in the South Pacific, has now had major impact in three important therapeutic areas. I can also show you the timeline of the application of rapamycin in the clinic. I like to show this to give you a feel for how recent much of this has been. So it was approved by the FDA for immunosuppression in 1999, about 20 years ago. It was then approved for use in stents or treatment of restenosis shortly after that. And then more recently, in 2007, it was developed as an anti-cancer drug. This is Pfizer's version, which is called Toricel. And then this is the Novartis version called Afinitor. So it has had impact in the clinic in the treatment of three major indications. So how did we get started in this area? Well, for us, it started in the late 1980s when a very good postdoc joined the lab by the name of Joe Heitman. And Joe Heitman was an MD, PhD, and given his medical background, was interested in how this new class of drugs, this new class of immunosuppressive drugs, which made transplantation surgery possible, he wondered how they worked. Another fortuitous circumstance was the fact that Rao Mova, who was a group leader at a local Basel company by the name of Sandoz, but now known as Novartis, and the fact that Sandoz, a major interest of Sandoz was immunosuppression. They had a major drug on the market at that time, which was used for immunosuppression. And of course, Rao was interested in trying to understand how these immunosuppressive drugs worked as well. So Joe teamed up with Rao, and it was actually through Rao that we were able to obtain rapamycin. Rapamycin was not generally available at that time. You could not buy it commercially. And what Joe and Rao did, which was a brilliant idea, was to use yeast to try to understand the mechanism of action of rapamycin. Now, this was a bit of an outlandish idea because what you have to realize is that that rapamycin rather was being developed for use in humans. And of course, all other groups who were studying this were trying to understand the mechanism of action of rapamycin in mammalian cells. But Joe and Rao had this idea to use yeast, because if you use yeast, you could use genetics to try to understand how this drug worked. And in those days, you could not do genetics with mammalian cells like you can today. Of course, everybody thought giving this human drug to simple unicellular yeast cells was a very irrelevant thing to do. But what Joe and Rao remembered, and what many others had forgotten, is that rapamycin evolved as a secondary metabolite in bacteria to act on fungi in the soil, and that in fact rapamycin had originally been isolated as an antifungal. So it had evolved to work on cells like yeast, and they therefore decided to exploit yeast to understand how rapamycin works, which was getting back to the roots of rapamycin. So what Joe and Rao did was a very simple experiment. They made some petri dishes with agar in them and spread rapamycin on top of these plates, and then plated a large number of yeast cells and incubated these plates for a couple days. And if there had been no rapamycin on this plate would have been covered with a thick lawn of yeast. But in this case, the rapamycin killed the vast majority of the yeast cells which have been plated here, and only a few spontaneous rapamycin-resistant mutants appeared on the plate. Joe then picked these individual mutants, streaked them out, purified them, and did more genetics on them to characterize mutants. You want to understand what genes are mutated, how many genes are mutated, and which genes are mutated. And what he found was that the vast majority of them, we found that the mutants were defective in any one of 3 different genes. The vast majority of the mutants were defective in what Joe had already characterized as the FPR1 gene based on his earlier work in the lab. And not only were most of the mutants defective in this gene, but these mutations were also recessive. In addition, Joe had isolated mutants defective in 2 other genes. These mutants were not only rare, but they are also dominant. And he called these, and since these were new mutants, unlike the FPR1, he called these Tor1 and Tor2 for target of rapamycin. So it was at this stage then that Joe left the lab, finished his postdoc, went back to New York to finish his medical studies, and we were left wondering, what do these genes encode? Why do we have mutations in 3 genes to give resistance to one drug? And why are some mutations very common and some very rare, in addition to being recessive versus dominant? So to answer all these questions, another student in the lab joined the project. probably the best student I've ever had in the lab, Jeanette Kunz, who you all know. So she decided to clone the genes, sequence the genes, and then knock them out and see if they are indeed to see if these genes indeed encode the product, the target of rapamycin, or code the protein which rapamycin binds. She was able to answer these questions of why there were 3 genes and some dominant, some recessive, and that relates to the mechanism of action of the drug. So here's rapamycin again, now drawn in a space-filling configuration. It's a very lipophilic molecule, so it simply diffuses across the plasma membrane. It doesn't need any specific transporter, which when mutated could confer resistance. And once inside the cell, it by itself does nothing. It must first form a complex with another protein called FKBP. This is a very small protein, 12 kilodalton protein. And it's then this protein rapamycin complex that binds directly to Tor, and that is then over here. Tor is a very large protein compared to FKBP. It's about almost 300 kilodaltons in size versus the 12 kilodaltons of this protein. So this is a very small portion of the entire Tor protein. It is what's known as the FKBP rapamycin binding domain. So whereas FKBP is completely essential for drug action, it's not essential for viability of the cell. So the cell can lose its protein and not suffer for that. So this is why any simple loss of function mutation in the FKBP encoding gene, which is FPR1, would confer complete rapamycin resistance, and such mutations would be recessive. So that explains why most mutations were in the gene encoding this protein. Tor, on the other hand, is completely essential for cell viability, unlike FKBP, and as a result, it cannot tolerate varied mutation if the cell is to survive. Indeed, every single mutation we identified, which mapped in the Tor gene, fell in the exact same codon, which specified a key contact site between Tor and rapamycin. It's actually a codon in this alpha helix, which contacted rapamycin. And of course, the effect of these mutations was to prevent the binding of FKBP rapamycin to Tor without otherwise affecting Tor activity. And these mutations were dominant because the wild type, the mutant version of Tor could still confer rapamycin resistance in the presence of a wild type copy of Tor. And they're rare because they're all confined to a single codon. So Jeanette answered these questions of why three genes and why some rare and some common. But what is Tor? What do the Tor genes encode? And Jeanette also answered this question by sequencing the genes. Of course, you had to do this all by hand in those days. And what we learned upon sequencing of the Tor genes was that the 2 Tor genes are very similar, 70% identical, and they also turned out to be the founding members of this novel class of atypical protein kinases, serine-threonine protein kinases, which we call PIKs, for PI kinase-related protein kinase. Now, every member of this class looks like a PI kinase, in other words, looks like a lipid kinase, PI4 kinase or PI3 kinase in particular, but none has actually been shown to have lipid kinase. This is something that Jeanette worked on very hard after she'd cloned and sequenced the Tor genes to try to understand what its lipid kinase activity might be, but in fact, it had no lipid kinase activity, despite the fact that it looked like a lipid kinase, but in fact turned out to be a protein kinase. All these are protein kinases. None has been shown to have lipid kinase activity. And to this day, we don't know why they look like lipid kinases. Since this early cloning of Tor in yeast, It's now been found in all other eukaryotes. It was a year after the cloning in yeast. It was cloned from mammalian cells, and this is part of the work that Sarbasov was involved in when he was a postdoc with David Sabatini at MIT. And we learned from this that Tor is indeed highly conserved all the way from yeast to human, and the name from yeast has now been kept for Tor from all other organisms, including flies, worms, and plants, in addition to mammals, usually with a prefix to indicate what kind of organism the Tor is from. So mTOR stands for mammalian Tor or mechanistic Tor, mechanistic target of rapamycin. So this is where we were in the mid-1990s. We had this very large, highly conserved novel kinase, which we knew was the direct target of rapamycin. But we didn't know what its role in the cell was other than being the target of rapamycin. Now, a great deal of work from our lab and a number of other labs showed that the role of Tor is to control cell growth. It's in fact a central controller of cell growth. And this was most photogenically shown in experiments done by Tom Neufeld and Bruce Edgar, two Drosophila geneticists, fly geneticists. In this experiment, and what you're looking at here is the fat body of the fruit fly, which is equivalent to liver or fat tissue of the fly. And the experiment that Tom Neufeld did was to selectively knock down Tor in these two GFP expressing cells. And as you can see, they're much smaller than the wild type neighboring cells. Bruce Edgar did the inverse experiment. Instead of knocking down Tor signaling, he hyperactivated Tor signaling. And again, you see that in these GFP expressing cells, and the cells become much larger than the neighboring wild type cells. So the obvious conclusion from these experiments in flies is that the role of Tor is to control cell growth. This was largely a confirmation of what we'd already learned in yeast cells. Now, Tom Neufeld took this experiment one step further. He isolated a Tor mutant fly in which all the cells of the entire animal are now defective for Tor. And it's important to point out that this was not a complete loss of function of Tor, because if it had been, this fly never would have been born. So it's what's called a hypomorph, a partial loss of function of Tor. It's actually a mutation in the promoter of the Tor gene, which reduces Tor activity to 25% normal levels. In any event, when Tom Neufeld isolated this mutant, as you can see, it turned out to be smaller but perfectly proportioned. And then Tom asked the question, why is this fly smaller? Is it smaller because it has smaller cells, but the normal number of cells, or is it smaller because it has normal size cells, but then fewer cells? And the way you can answer this question is by looking in the wing, which is a plane of cells, so we could easily count the cells and measure their circumference. And what he found was that this smaller fly had more or less the normal number of cells, but each individual cell was smaller. So this told Tom Neufeld that Tor controls not only cell size or cell growth, but also thereby controls size of organs and ultimately size of the whole animal. Now, to my mind, this is an absolutely astounding result, because to make a fly like this is a program in 4 dimensions. And this program seems to have been rewritten in all 4 dimensions by a single mutation in the genome, in this case, in the promoter of the Tor gene. I never would have thought this would have been possible until I'd actually seen it. You can think of all the regulatory and metabolic circuits which would have to be rewired to generate this smaller but well-proportioned fly. Now, it's also important to realize that Tor controls growth in many other physiological contexts, oftentimes independent of cell division as unlike what you saw on the previous slide with the fly. Here's some examples. If you fast or feed, your liver changes in size, and that's not because you acquire more liver cells or lose liver cells, but it's because the cells you have become larger or smaller. One of my favorite examples is muscle. If you exercise, you know your muscles get larger, and you don't acquire more muscle cells, but your existing muscle cells get bigger. Interestingly, in this context, Tor is actually broken into popular culture, gone mainstream. You can see that here. This is a dietary supplement, which you used to be able to buy on the internet. It's a dietary supplement for bodybuilders. And you can see if you take, it's on the label, it says mTOR and FOXO and many of these official scientific terms. What's actually in this flask, I don't really know. It's probably simply leucine for reasons I'll get to later. But it gets worse than that. We all like mind-blowing results. When Tor signaling, you can even get something called max Tor, again, for bodybuilders to get these mind-blowing results. Not the kind of mind-blowing results that we had in mind, but mind-blowing results apparently nonetheless. Okay, back to more serious matters. In the case of the central nervous system, there are very dramatic changes, not only in cell size and cell shape, but Tor also upregulates. Tor is also responsible for this. And someone has even shown that queen bees are larger than worker bees, for example, due to upregulation of the Tor in the larvae, which give rise to this bee. So the worker bees feed a special royal jelly to the larvae, which develop into queen bees. And the special super nutritious royal jelly is what upregulates the Tor signaling pathway in those larvae to induce development of a queen. So how does Tor, so Tor controls growth very broadly in terms of organism and broadly in terms of physiological context. Essentially any case in nature where you see growth change in size, either at the cellular level, organismal level, there's a very high likelihood that Tor signaling is responsible. So how does Tor do this? How does Tor control cell growth? Well, again, work from a number of labs, including our own, show that Tor controls a large number of cellular processes which collectively determine mass accumulation and thereby cell size. And these processes can be subdivided into two groups: the anabolic processes which Tor activates, and the catabolic processes which Tor inhibits. So what Tor does to control cell growth is it balances these opposing forces of synthesis and degradation, and it does this, and this is important, in response to nutrients. So in very simple terms, Tor is what makes us grow when we eat. Now, how does Tor control all these different processes? Well, there are effector signaling pathways which emanate from Tor, and these pathways intersect with key proteins involved in all these different processes to control them. In some cases, we know what those effector pathways are and what those key proteins are, but in many cases, we still don't know. But here's the picture that had emerged by 2000, and that is that there are in fact two Tor signaling pathways. Therefore, we now call it Tor signaling network because it's more than a single pathway. And this is in yeast. We're still in yeast. You can tell that because there are two Tor genes. Something I didn't mention earlier is that yeast is unique in having two Tor genes and therefore two Tor proteins. All other eukaryotes, all the way from insects to worms to plants to mammals, have a single Tor gene and a single Tor protein. So this is yeast. You can tell by the two Tor proteins. And the two Tor proteins allowed us to discover that there are in fact two Tor signaling pathways because we could define them by differential requirement for these two different Tor proteins. So one pathway, which we often call the Tor2 unique pathway, because only Tor2 can signal through this pathway, controlled organization of the actin cytoskeleton. And this determines how new mass is laid down in the cell. And therefore, we view this pathway as mediating the spatial control of cell growth. The other pathway, which we called the Tor Shared Pathway, because either Tor can act redundantly in this pathway, controls these processes which lead to actual mass accumulation, these anabolic processes, and this is what is responsive to nutrients. So we view this pathway as mediating the temporal control of cell growth. So we believe the logic of having two separate signaling pathways is that they integrate spatial and temporal control of cell growth. And of course, these two parameters of cell growth have to be integrated for coordinated cell growth to occur. So this is where we were in the year 2000. We had still many questions. What determines the specificity of the two Tor proteins? Why can Tor2 signal through both branches, yet Tor1 only through this branch? Also, something we learned and it puzzled us was FKBP rapamycin could bind directly to Tor to sequence this pathway, but seemed not to be able to bind Tor2 in this pathway, although it's the same Tor2 protein here and here. So why could rapamycin inhibit here, but not here? Now, to answer these questions, we decided to take a biochemical approach, in particular to purify the two Tor proteins using a purification scheme which would be gentle enough to isolate interacting proteins, and therefore would give us some indication of the events which are occurring near Tor itself. There's the work of a postdoc in the lab at the time by the name of Robbie Loweth. He did, in fact, isolate two Tor complexes out of yeast cells, which he called Tor1 for Tor complex 1 and Tor2 for Tor complex 2. And once he had these two complexes purified in the test tube, this allowed him to sequence the proteins and make up these various subunits of these two complexes. And once he had that information, he could then isolate the genes encoding all these proteins. And once he had that, he could then knock out these genes to study them genetically. And when he did all that, he found that these two Tor complexes corresponded to these two previously identified Tor signaling pathways. So this was a major breakthrough for us because, first of all, it said that Tor is in fact two structurally and functionally distinct kinases based on what complex it's found in, but also answered these questions that I posed earlier. Why can Tor 1 and 2 signal redundantly through this pathway? And the answer is, of course, that either Tor can assemble into this complex, but only Tor 2 can assemble into this complex. And we also learned that FKBP rapamycin can inhibit here, but not here, because FKBP rapamycin can bind this complex, but for some reason was unable to bind this complex over here. We now know the reason why it can't bind this, but I'll come to that later when we talk about the structure of these two complexes. So I also have to say it was a very gratifying moment in the lab when we showed these two complexes corresponded to these two previously identified signaling pathways. There was a convergence of many years of genetic research which identified or delineated the two signaling pathways and years of biochemistry which identified the two complexes. It was a convergence of these two areas of study into what is a unified model of Tor signaling, which we sometimes call the two-branch, two-complex model of Tor signaling. And of course, the genetics and the biochemistry cross-validated each other. So we have a very high level of confidence in this model of Tor signaling, network. So this is where we were then in the year 2002. And having discovered these two complexes in yeast, we then wondered if they are conserved mammalian cells. And if we looked in what was available for the mammalian genome sequence, there was no complete mammalian genome known at that time yet. We looked for orthologs of the proteins which made up the two complexes from yeast. We looked in the, so we could find orthologs in the mammalian genome database. And we did find some proteins which had some degree of identity, which suggested that the two complexes, like Tor itself, might be conserved all the way from yeast to human. So we started working on mammalian cells to see if, in fact, these orthologs of these proteins make up Tor complexes in mammalian cells. And so but at this stage, mainly the laboratory of David Sabatini, Kunyang Guan, and Kazu Yonezawa showed that indeed the two complexes are conserved, whether in mammals, where they're known as mTORC1 and mTORC2. They phosphorylate many of the same proteins in mammals, or orthologs of the same proteins that they phosphorylate in yeast cells. And they also respond to nutrients, amino acids in particular, in mammalian cells. So the picture that emerged here is that the entire, this Tor signaling network is a primordial or ancestral signaling network, which has been conserved throughout eukaryotic evolution to control this very fundamental process of cell growth. Now, one exception to that statement I just made is this part of the network up here, which is the insulin or growth factor signaling pathway. This evolved much later than Tor itself. This is not present already in unicellular organisms like yeast. This evolved with multicellularity and was then grafted onto the more primordial Tor signaling network. And the reason for that is that cell growth control in multicellular organisms, where this evolved, is more complex than cell growth, control of cell growth in unicellular organisms, because in multicellular organisms, metazoans, it's critical to control the growth of every cell in the body with every other cell in the body, such that our organs end up being properly proportioned. And that's then what this growth factor signaling pathway provides. It provides a global input on cell growth control to control growth over a whole body plan. So in metazoans or multicellular organisms, we know we have the global growth factor input combined with local nutrients, amino acids, and now we also know local energy in the form of ATP. So once we have these three factors present, growth factors, nutrients, and energy, the entire Tor signaling network is upregulated. All these anabolic processes are stimulated and cell growth ensues. Now, another fascinating aspect of the Tor signaling network is the unusually large number of diseases to which it had been linked. And this all emerged as other scientists started cloning the genes defective in various diseases and discovering that they all affected proteins that made up components of the Tor signaling network. So many of these proteins were not even known to be functionally linked until they were mapped onto the Tor signaling network. And what all these diseases have in common is they're all characterized by inappropriate or ectopic cell growth. And this can be malignant forms of cell growth or benign forms of cell growth, but in all cases, cell growth, which underscores the notion that Tor is a central controller of cell growth. More recently, Tor has been implicated in another set of diseases, the so-called metabolic disorders. This can be obesity. We know that chronically high levels of circulating nutrients can upregulate Tor even in the absence of growth factors, and this can lead to adipogenesis, differentiation of fat cells, which can lead to obesity. And this can in turn lead to diabetes. But there's a more direct link to diabetes. We know there's a famous negative feedback loop from Tor to IRS of the insulin signaling pathway, such that high levels of nutrients or a poor diet can lead to phosphorylation of IRS. This would inhibit signaling through the insulin pathway and confer insulin resistance, which as you probably know, is one of the hallmarks of diabetes. So this provides a molecular link between diet and insulin resistance. In this case, some have actually proposed using rapamycin in a fourth therapeutic area, treatment of diabetes, because if you inhibit Tor, you would inhibit the negative feedback loop and you restore signaling through the insulin pathway. And needless to say, this would then restore insulin sensitivity. Nobody, to my knowledge, has actually developed rapamycin as an anti-diabetic because there are a number of other complications which would prevent that. But something giving credence to the notion that it could be possible is that the world's most commonly prescribed anti-diabetic, metformin, works at least in part by downregulating mTOR signaling via AMPK, which is a negative regulator of mTOR signaling. Now, another finding which created lots of excitement in the Tor signaling pathway was the fact that it controls lifespan. This was first shown in worms about 20 years ago, then in yeast and flies. In this paper in 2009, it was shown for the first time in mammals. In fact, this is the first pharmacological extension of lifespan in mammals. And this created lots of excitement because it's been known since 1935, so 85 years ago, that dietary restriction or caloric restriction, in other words, eating less, extends lifespan. But nobody understood the molecular basis of this phenomenon. And with this discovery here, it seems to provide an explanation because we know Tor is a nutrient responsive kinase, it's a nutrient sensor, and by inhibiting Tor, what you're essentially doing is tricking the cells into thinking there are no nutrients, makes the cells unable to sense nutrients, and this then unleashes all the same metabolic changes you'd see if you indeed ate less. So it appears that the dietary restrictions extends lifespan by downregulating Tor signaling and all the accompanied changes in metabolism that occur as a result. At least that is the current mechanism, current model for how dietary restriction extends lifespan, which is, needless to say, also causing a lot of excitement. So this is where we were about 10 years ago. We then we had a very good idea of how Tor controls growth of individual cells. But then a new problem arose, a new interesting problem arose, and that is how does Tor actually control growth of the whole organism? Not just of individual cells, but a whole multicellular organism. And this challenge was inspired by work of Pierre Leopold, who was a French fly geneticist, and much like the work of Tom Neufeld, which I showed you before, Pierre was looking for mutant flies which had a growth defect. And he isolated a mutant fly, which was smaller, like the Neufeld fly. And this fly had a mutation in a gene encoding an amino acid transporter, which was specific to this one tissue of the fly. So these are different tissues of the fly. And this mutation he called SlimFast, and the tissue is the fat body, the same tissue I showed you before. And since this amino acid transporter was specific to this one tissue, amino acids didn't get into the cells of this one tissue. And as a result of that, Tor, or Drosophila Tor in this case, was not activated in cells of the fat body. And as a result of that, as you might expect, the cells were smaller and the fat body was smaller. Now, bear in mind, this transporter is specific to this tissue, so these other tissues have their own transporters, and amino acid transport into these other tissues would not be affected. Nevertheless, all these other tissues were also proportionally smaller to match the size of this one small tissue over here. So what this told us was that there is Tor-dependent signaling between tissues to coordinate growth over a whole body plan to ensure that the different tissues are properly proportioned. If you limit one tissue, you can control the growth of the other tissues to match that one tissue. So this is what we wanted to then study after we'd more or less elucidated how control of a single growth is controlled. We want to understand how growth of the whole animal is controlled. To do that, we didn't want to work in flies, but in mice. So we started working on mice. And what we decided we had to do was to understand the function of TORC1 and TORC2 separately in every tissue of the body, to understand how all the tissues are communicating with each other in a TOR-dependent manner to control whole body growth. So we made mice which were defective for either Richter, which is an essential and specific subunit of TORC2, or we made mice which were defective in Raptor, which is an essential and specific subunit of TORC1. And we made these mice conditional knockouts, so we could knock out TORC1 or TORC2 individually in any specific tissue we wanted. Then the question arose, which tissue should we look at, because TOR exists in all tissues? So we decided to look at the metabolic tissues, fat, liver, muscle, brain, and intestine, because these are the tissues which are most insulin sensitive, most nutrient sensitive, and are the tissues which control energy homeostasis in the body. And these are precisely the three inputs which control TOR signaling, growth factor or insulin, nutrients, and energy. So we thought that TOR would have a particularly important or interesting role in these tissues. So we did that, and let me just give you one very quick glimpse of what we saw when we knocked out TOR in fat. So this is a mouse which is lacking TOR complex 1, specifically in the adipose tissue or the fat tissue. And both these mice, here's a wild type mouse, both these mice were put on a high fat diet. And as you can see, this mouse was completely protected from obesity, unlike the wild type mouse. Furthermore, we found this mouse was metabolically healthier than this mouse. It had lower levels of insulin, lower levels of cholesterol, which then harkens back to this notion that TOR controls aging. In the experiment I showed you before, where they fed rapamycin to mice to extend lifespan, rapamycin would have inhibited TOR in all tissues. But then that begs the question, if inhibiting TOR extends lifespan, is there a particular tissue in which you would have the beneficial effect of inhibiting TOR signaling? In this mouse, we've inhibited TOR signaling in this one specific tissue, fat, adipose tissue, and we get a healthier mouse. We never did the experiment, but the prediction now is that this mouse would live longer than the wild type mouse, and therefore inhibiting TOR in adipose tissue is particularly important for extending lifespan. So what happens when we knocked out TOR complex 2? Here we had a surprise. We knocked out TOR complex 2, the mice turned out to be larger than the wild type mouse. And that let us discover this new axis of signaling between tissues that control insulin-like growth factor and insulin levels in the bloodstream. So we found out when we knocked out TOR complex 2 in adipose tissue, levels of these two growth hormones increased in the bloodstream. That's because we failed to turn off IGF synthesis in the liver or insulin production in beta cells of the pancreas. So we think TORC2 in adipose tissue's role is to control IGF insulin signal production in these two different tissues as a way to control IGF insulin homeostasis. And that's due to the fact that TORC2 itself responds to IGF and insulin. So if these levels get too high, they activate TORC in adipose tissue, which turns down their synthesis to restore insulin and IGF to normal levels. In the absence of TORC2, these growth hormones increase in concentration in circulation, which leads to this systemic growth, like you see over here. So we've made interesting observations essentially in every tissue we look at for loss of TORC1 and TORC2. These are just two examples. Now, to end, let me go more molecular. So we spent many years and are still trying to understand how the physiology and pathophysiology of Tor signaling. But at that same time, we're also looking at Tor more at the atomic level, trying to understand the structure of the Tor complexes. A few years ago, we were able to determine the structure of Tor complex 1 at about 5 angstrom resolution. And what we learned is that Tor complex 1 is a dimer. There are 2 Tors. This is Tor here and a second Tor here. This is Raptor, the protein whose gene we knocked out in the mice. And this is the third subunit of Tor complex list 8. So Tor itself turns out to be a dimer of heterotrimers. Since it's a dimer, it has 2 catalytic sites. This cleft here is the kinase catalytic site. It has 2 of them. And we also determined the structure of Tor complex 1 bound to FKBP rapamycin. So if you keep your eye here and here, you will see FKBP rapamycin binding. There they are. So this explained to us why you need both FKBP and rapamycin to inhibit Tor complex 1. Explains why rapamycin alone is not sufficient. Because the mechanism of action of inhibition is that rapamycin glues FKBP onto the lip of this catalytic cleft to form a lid, to close or to cover up this cleft to block access of substrates to the catalytic site here and here. And of course, rapamycin alone, which could still bind TORC1 in the absence of FKBP, is simply not bulky enough to form this lid to block access of substrates. So we also learned that Tor binds its substrates through rapamycin, which are then presented to the catalytic site here and here. But this is still ongoing work as well. What you don't understand from looking at this figure is how big Tor complex 1 is. It's an exceptionally large kinase. So to give you a feel for the size of Tor complex 1, let me show you what it looks like next to something you will recognize. So here is again Tor complex 1, now drawn in a ribbon configuration. If I'll stand it up, and here's a ribosome. So Tor complex 1 is as big as a ribosome, much bigger than I expected it to be, even though I knew it was about a megadalton in size. But you should be impressed not only by the size of this kinase, but probably also by the fact that how much more dense a ribosome is compared to Tor complex 1. And I think this lack of density over here reflects how much we still have to learn about Tor complex 1. Once we know the structure of Tor complex 1 bound to all its regulators, such as Reb and RAG, this will probably look as dense as this. And of course, many labs are trying to, are determining those structures right now, including David Sabatini's lab at MIT. Okay, so that's Tor complex 1. Even more recently, we discovered or determined the structure of Tor complex 2 down to 3.2 angstrom resolution. And what we learned is that Tor2 is also a dimer, but there are 4 subunits rather than 3 in Tor complex 2. So this is a dimer of a heterotetramer. Tor itself, mTOR itself, has the same structure in both complexes, but then it's decorated by its specific subunits in different ways, which still bind Tor in the same region, which is why these 2 complexes are mutually exclusive. Now, I think the most interesting thing we learned from the structure of Tor complex 2 is why FKBP rapamycin cannot inhibit Tor complex 2 like it can inhibit Tor complex 1. So this is this is FKBP rapamycin here and here. What we learned, and finally solving this open question since we'd had since 2004, is that the reason that FKBP rapamycin can't inhibit Tor complex 2 is that Richter, the subunit Tor complex 2, masks the rapamycin FKBP binding site in Tor such that this FKB rapamycin cannot bind Tor2 to inhibit it. Okay, I think I've come to the end of a of the story of Tor. I've tried to give you a feel for its discovery, the genetics, the physiology, pathophysiology, and atomic level resolution of Tor signaling. Okay, these are people in the lab. Unfortunately, I was not able to discuss any of their work except the more recent structural work, which was done in collaboration with Tim Meyer here at the Biozentrum, and then at Bonn, the ETH in Zurich. Okay, I'm happy to take any questions if you might have them. I'll have to stop sharing if I want to do that.
Do you want to handle the questions yourself, Mike?
I can handle. Okay, first question. I suppose all of you can read the questions as well, right? We know that mTOR is responsible for avoiding apoptosis, growth, cell proliferation, metabolism. Well, in some cases it promotes those, but in any case. However, this signaling pathway is said to have several tissue-specific functions. What exactly can these features affect? So what are there in tissue specific, what tissue specific factors are there which may confer specific functions on Tor signaling? There are actually not very many Tor specific tissue specific functions of Tor. Tor controls growth in essentially all cells. There might be some specific metabolic targets or specific genes which it activates in one tissue versus another tissue, but the overall function, the primary function of Tor, which is to control general metabolism, general cell growth, is conserved in all tissues, generally by signaling through the same effector pathways to control the same processes such as protein synthesis, lipid synthesis, and nucleotide synthesis, and suppression of autophagy, inhibition of autophagy. And how these small differences might be created might be, for example, it could turn on transcription, Tor could turn on transcription of one gene in one cell, but not another side because a co-transcription factor might not be expressed in one tissue versus another. The message I want to leave you with is that, in fact, Tor's function is more or less conserved. Its general function is conserved in all tissues, not only in all tissues, but in all organisms. Okay, next one. In connection with the COVID pandemic, do you think that vaccination against COVID is a panacea for the disease, or the body will be able to cope with the disease on its own? Well, I think this is a general question of vaccination. Vaccination is extremely important. I encourage everybody to get vaccinated. I myself have been vaccinated, and I don't think we will be able to live with this disease until we get vaccinated, or at least until we develop immunity. It'll continue to evolve and continue to throw off variants, which might get deadlier and deadlier. So I think it's important to get vaccinated, and I think it's important to get vaccinated as soon as possible so we can outrun all the variants which are popping up. I don't think we will ever be able to cope with this disease. My feeling is if we ever are able to cope with this disease independently of the vaccine, it might take a few million years of evolution, but and that's still a bit too slow for us. Okay.
So Mike, I don't know if we have some more time, but there are many diseases that are kind of linked to Tor. So what are you wanting to do in the future? What are your future plans? Do you know a specific disease or?
Well, yes, we are focused on, this is probably the major area in our lab right now. We think we've have a pretty good understanding of what Tor does in health. And now the natural progression is to see what is Tor doing in disease.
Yeah.
And this is what we're focusing on. And there we're focusing on two very major areas, cancer and diabetes. At the very moment, if I'd given you a lecture today on the current work in the lab. What I've told you about is probably a project on diabetes. How Tor signaling in fat is altered in a high-fat diet to lead to hyperglycemia, to high blood glucose levels, which are, of course, what causes diabetes.
Yeah, I think it's a no-brainer, huh?
Yeah, it's a no-brainer.
Teaching, of course.
It takes brain to figure it out, but not the
Yeah.
idea to go there.
And there's actually another question, which may be our last. I was asked if Tor changes over generations, so evolution of Tor.
Well, it's very similar between yeast and mammals. That's about 2 billion years of evolution right there. So I think in very broad brushstrokes, it has not changed very much since from yeast all the way to mammals. Fortunate for us, because that allowed us to discover it in yeast. But no, I think it's like histones. They perform such an essential function that once Mother Nature discovers something, tinkers around and develops something through evolution, and it works, and it's something important, it doesn't change. It's locked in. So I think Tor is an example of how something was not easy to make, but it does something so important it can't tolerate very much change at this stage.
And I think I just have the last question. Maybe there is this other pathway. So Tor controls the size of the cell, and there is the hippo signaling pathway that counts the cells. Do these two pathways actually talk to each other directly?
Yeah, there have been a few publications in which the two pathways seem to talk to each other. For example, YAP, which is the downstream transcription factor in the hippo pathway, seems to control expression of P10, which is a tumor suppressor upstream of Tor. And there are a few cases, specific molecular examples of how these pathways crosstalk. When hippo was discovered, much more recently than Tor was discovered, I got very excited about that because, again, this answers the question of how do we coordinate cell number with cell size to determine size of the whole organism. And I expected many very obvious crosstalk examples to come out right away, but it has not been the case. There are some, but not as much as I expected. It's a good question.
Well, I think we're pretty much within the time still. So I want to thank you so much for your fascinating talk. And this really has been the most interesting scientific topic I worked on in my career so far. So
we miss you in the lab too, Jeanette.
Good memories. The bad memories have gone into. So I hope we see you at some point here. In Nur Sultan, and actually have a talk to you personally. So
I hope so too.
Thank you so much, and I also want to thank the audience for the questions, and I want to encourage you to maybe contact Dr. Michael Hall in the future if there are any more questions of this, any interest in working at the University of Basel. And I want to finish here, and I think there's going to be a recorded question by another fellow Swiss, Wüthrich, Dr. Wüthrich, who will follow this talk. So thank you very much, Mike, and stay safe, and we hope to see you here. Thank you.
Thank you. Bye-bye, Jeanette. Good seeing you.